See, look at the board again. So in the previous class, we said that in the paper 2, right? In the paper 2, we're having the syllabus we called as one is the genetics, another is the plant breeding, another is the molecular the breeding, another we're having the total syllabus we called as biotechnology. And within this genetics and plant breeding, then we're going to apply the statistics in the name of the bio statistics to find the whether the developed variation is a significant or non-significant like that. So here today onwards we'll be discussing exclusively the fundamentals of the genetics. So especially in the section A of the paper 2, section A of the paper 2. So in the UPSC, these are the following the syllabus we have in the genetics here, right? One is the cell structure and functions of the cell and cell cycle here, right? See, so within the cell, right? Within the cell, we're having the different kind of the components or organelles can be possible. Within the cell, especially in the case of the plant cell, we're having the mitochondria, protoplasma, cell membrane, cell wall, and nucleus, and other Golgi bodies can be possible, right? So there will be discussing all the organelles and their functions in the plant growth and development. And apart from that, we're having the syllabus we called as cell cycle. So within the cell cycle, we're having the two phases, right? In the case of the division, one is the every other term interphase, right? Within the cell cycle, we're having the two phase. One is the phase of the interphase, we say. After the completion of the interphase, then we're having the mitotic cell division. Either it can be mitotic or meiotic cell division, right? So in the cell cell cycle, right? And generally, so in every plants, not only the plants, in all the living organisms, we're having the cycle we called as some are the interphase. Once after completing the interphase, then it is entering into the either mitotic or meiotic cell stage can be possible. So there are the two stages here. One is the interphase, another is the mitotic phase here, right? Within the interphase, we are having the what we called as G1 phase, G2 phase, and S phase can be possible. G1 phase, G2 phase, and S phase can be possible. And after completing the G1, S, and G2 phase, then it is entering into the M phase. M matlab either it can be mitotic or meiotic. That is the one process of the cell cycle can be possible. So for example, in the case of the plants, in approximately, right, it takes 31 hours to complete the one cell cycle. It takes 31 hours to complete the one cell cycle. So from the, it is starting with the interphase. After that, we are having the G1 phase, S phase, and G2 phase. G1, S phase, and G2 phase. Once after completing the G2, then it is entering into the M phase here, mitotic or meiotic. So once after completing the mitotic, then it's called as one cell cycle. So in the experimentally, it is observed in the case of the legumes, in the legume crops we called as Pisum sativa. Pisum sativa, there experimentally it is observed that minimum 31 hours it takes for the one cell cycle, right? So for example, even in the human also, whenever having some injury, skin, right? So injury on the skin, over the 2 to 3 days, there is a recovery of the skin can be positive. There is a one cell cycle in the human genetics here, right? So that is the one syllabus we have here, right? One is the plant cell, how the plant cell is differ with the animal cell, and within the plant cell we are having the various kind of the organelles. Organelles matlab components, cell wall, cell membrane, protoplasma, nucleus, Golgi bodies, right? So apart from that, we are having the cell cycle. So we can expect the 10 marks question on the case of the cell cycle. So there we are having the two phases. One is the interphase followed by the mitotic or meiotic. Within the interphase, again we are having the G1 phase, S phase, and G2 phase. Once after completing the G2 phase, then it is entering into the mitotic cell division. Mitotic cell division. Within the mitotic, again we are having the subphases: prophase, anaphase, telophase. Every other term. That is the sub stages here. Then apart from that, we are having the synthesis, structure, and functions of the genetic material. Genetic material matlab either it can be DNA or RNA. So there you will be discussing the functions of the DNA, structure of the DNA, and how the synthesis of the DNA is going to take place. Synthesis matlab the kind of the biosynthetic pathway for the production of the DNA particles. Then apart from that, we are having the loss of the heredity. So loss of the heredity is a very broad concept. So there we are having the Mendel law. Mendel law is one of the aspect which defines the laws of the heredity. Remember point. For example, we are having the Mendel law, linkage theory, right? Chromosome or cytoplasmic inheritance, right? So laws of the heredity is a broad concept. In that cases, we are having the Mendel law. So main objective of the Mendel is to study the inheritance of the character from the one generation to the another generation. That is the one theory view. Another is the linkage theory. Linked group of the genes will be inherited from the parent to the progeny. Third one, we are having the cytoplasmic inheritance. So cytoplasma is present only in the female parent but not in the male parent. So that is also one of the aspects to define the laws of the heredity. Remember point. So laws of the heredity is a very broad concept. Within that there are the many theories we have to define the heredity. Laws mean framing the rules and regulation. To study the how the inheritance of the character is going to take place from the one generation to the another generation. That is the syllabus we have in this cases here, right? So especially within that we are having the cytoplasmic inheritance, Mendel laws, another is the chromosomal linkage theory. Then apart from that we are having the chromosome structure and chromosome aberration. So here we can expect 10 or 15 marks here. 10 or 15 marks we can expect in the case of the chromosomal aberration. Aberration matlab whenever the organism is exposed to the different kind of the environmental factors or mutation, right? Whenever you are having the mutations in the given chromosome, right? Changes in the chromosome can be possible. Changes matlab either it can be structure of the chromosome, either can be number of the chromosome, right? Structure or number, right? So both in both the sense there is the aberration. Aberration matlab deviation. Deviation in the normal chromosomes can be possible whenever the organism is exposed to the different kind of the agent. So that we call this aberration, we say. Then we are having the linkage, 10 marks question. Again we are having the chromosomal crossover, right? So at the time of the pairing time, right, from the normal chromosome to the recombinant chromosome, exchange of the some kind of the somatic segment can be possible from the normal chromosome to the resultant chromosome. Due to the crossover also, development of the new gene combination can be possible or not. Due to the crossover from the normal to the recombinant one, their exchange of the gene can be possible. That led to the variation in the biological population can be possible. So based on the such variation, development of the new character in the agriculture can be possible. That is the one advantage as well. Then, so here also we can expect 10 marks and 10 or 15 marks question. In the 2024, they asked a good number of the questions in the case of the aberration. Aberration matlab deviation from the normal structure of the chromosome. So normal structure or number, right? So there we are having the hyperploidy condition, polyploidy condition, addition, deletion, and inversion, translocation. So with respect to the structure, we are having the addition, deletion, inversion, and translocation. With respect to the number, we are having the aneuploidy and polyploidy condition. Then we are having the polyploidy condition. I think polyploidy we have discussed in. So it is a situation in which the organism is having the more than two basic set of the chromosome, right? Within that we are having the heteropolyploidy and allopolyploidy. We have its significance in the plant breeding. Then mutation is the most important topic. Again we can expect minimum 15 marks we can expect in the mutation. Mutation matlab sudden irritable changes in the given DNA sequence. In the given DNA sequence, some changes can be possible. That change either can be addition or deletion or duplication or inversion or translocation. We have all the possibility, right? And due to the mutation, sometimes it can be aberration in the number of the chromosome also can be possible. We have both, either it can be with respect to the structure of the chromosome or number of the chromosome, right? Then according to the mutation, whatever the variety we are developing, that variety is called as the mutant varieties. You remember in the plant breeding, in the mutation breeding, we said some table important mutant varieties in the based on the mutation breeding, right? Then we are having the what we call as 6 linkage, 6 influenced character, and 6 limited characters. Then we are having the 10 marks question we called as DNA fingerprinting. Apart from that, we are having another syllabus we called as cytoplasmic inheritance. Here, right? See, in the UPSC, this is the basic syllabus we have, right, in the agriculture perspective. So for the study purpose, we have divided the entire life science into the 5 subjects: genetics, plant breeding, molecular breeding, and biostatistics and biotechnology. Okay, so this is the basic syllabus will be there. Every year minimum to maximum 3 questions we can expect in the given syllabus it is, right? So in the case of the DNA fingerprinting, we said, right, I will remember the basic principle is that 90 is to 10% DNA. So 90% of the DNA will be remain same from the one organism to the another organism, but only 10% of the DNA, that 10% is available in the longer fragment or shorter fragment. Shorter fragment that will be inherited together from the one person to the another person. One person of the parent to the progeny. 90% will be remain similar. Okay, based on the 10% DNA, we are comparing the sample DNA with the hybridized DNA to confirm the identity of the individual. Identity of the individual it is. Right. So here, apart from that, so today, so we'll be discussing minimum 15 to 20 marks we can expect in the case of the mutation and their role in the crop improvement it is, right? So we said that what do you mean by mutation? Sudden heritable changes in the DNA sequence. Sudden heritable changes in the DNA sequence can be possible whenever the organism is exposed to the different kind of the environmental factors. Environmental factors over the period in the given living organism, some kind of the changes can be possible at the gene level. At the gene level. So in the given DNA sequence, so it can be addition or deletion or duplication or translocation of the character can be possible. According to the such changes, sometimes there is a new variation in the biological population can be possible. Based on the such variation, so improved varieties are developed in the agriculture. So output, whatever the output we are getting from the mutation, that output is called as the mutant varieties. Is called as the mutant varieties. Right. So here we will be discussing definition, right? So it is a more of the certain heritable changes in the given DNA sequence. So after the changes, the resultant phenotype will be same or different? Different. The phenotype will be totally different from the parent from the parent. Then how does mutation it occurs in the plant, right? Sometimes it is also called as important factors which is responsible for the mutation. How does matlab we are having all the biotic factors and abiotic factors collectively can lead to the changes in the DNA can be possible. Any idea what are the factors here which bring the mutation in the plants? One is the temperature, radiation, then pest and disease outbreak in the ecosystem, and what about migration from the one place to the another place can lead to the mutation or not? Migration and isolation of the species can be right. Any other apart from this? So we are coming shortly. So there are the many factors which can lead to the mutation in the plants can be possible. Then apart from that, so we are having the important features of the mutation. So mutation, so it can take place at any stage from the sowing to the harvesting, at any stage of the given life cycle. I'm taking small example in the plants from the sowing to the harvesting. In between any stage, mutation can be possible in the given life cycle. In the given life cycle, mutation can be possible at any stage. That is the one. And it is a universal or non-universal? Universal. In all the living species, the mutation can be possible. And don't you think so? It is a mutation is one of the factor in the evolution of the new species. So one of the ingredient is one of the ingredient which is highly responsible for the evolution of the new character in the plant breeding. It is right. Then apart from that, and within the mutation, sometimes it can be natural mutation, sometimes it can be induced mutation can be possible. Naturally occurring mutation, and even with the laboratory we have discussed on the mutation breeding. The plants will be exposed to the different kind of the chemical and radiation to generate the mutant character. Right? Sometimes it can be induced or it can be naturally occurring mutation. Sometimes within the mutation, sometimes it can be macro mutation or micro mutation can be possible. Macro matlab larger changes, micro matlab smaller changes. Sometimes mutation it can be forward mutation. Forward matlab going from the wild character to the present form of the alleles. Going from the wild character to the present form of the characters. And sometimes it can be backward mutation also can be possible. Going from the present to the past. Past to the present and present to the past. We have both the possibility here, right? And apart from that, so mutation it can be possible with respect to the structure or number of the chromosome also can be possible. Like that, there are the different type of the mutations can be possibilities, right? Then advantages and disadvantages of the mutation. Then important mutant varieties. Already we have discussed it in the plant breeding for the 10 marks question. Mutant varieties in the case of the rice crop, cotton, sugarcane, and other things it is, right? Then conclusion. So here, so we can expect 15 to 20 marks we can expect in the case of the mutation here. So here please write on this one. So we said that mutation it means certain heritable changes in the DNA sequence. The DNA sequence can be possible. So that kind of the change, either it can be structure of the chromosome or number of the chromosome can be possible. For example, with respect to the structure of the chromosomes, we are having addition of the chromosome or deletion of the chromosome can be possible, or sometimes it can be inversion of the sequence. Sometimes it can be duplication or translocation. We have all the possibilities in the changes in the given DNA sequence. Translocation. So whenever we are having the such kind of the changes, right, over the period there is a due to the crossing over, due to the crossover, exchange of the character from the original chromosome to the recombinant chromosomes can be possible. According to that, development of the new allele can be possible. Due to this mechanism, development of the new allele can be possible. Based on the new allele, right? So improvement of the new varieties. Improvement of the new varieties in the agriculture can be possible. Agriculture can be possible. So such varieties are called as the mutant variety species. The mutant, the variety species. So we said that in simple word we can say it is a kind of the changes in the given DNA sequence in the given DNA. So that change either it can be addition of the character, or deletion of the character, duplication matlab multiplication, right? So in the given DNA sequence we are having the ABC, and again ABC will be repeated 2 times in the given sequence of the chromosome. It is due to the crossover mechanism, so there is a new allele sometimes can be possible in the population. It is over now. So here let us take some examples here. So here we are having for example, these are the original chromosomes. So we are having the ABC and DE sequence in the original chromosomes. After the mutation in the resultant chromosomes, we are having these are the sequence can be possible. So what kind of the mutation it is? Original chromosome and the recombinant chromosomes here. So we are having the A, B, C, D, and here we have only A, B, and D. Any idea? It is a deletion. It is a deletion type of the mutation can be possibilities, right? Please write down the diagram. And here what type of the mutation it is? So here where the B is repeated 2 times, right? So it is called as duplicated mutation. And here in the third one, any idea? A, B, C, D. But in the recombinant chromosomes, we are having the ulta chromosomes. What kind of the mutation it is? Inversion. Inversion we say, okay? Inversion mutation. So it is a duplicated, another is the inversion mutation. So not only it is a structural mutation or numerical mutation. Structural, right? So it is a structural mutation. Numerical matlab, for example, 22 will become the 44. At some time it can be 44 to the 20 also. So we called as this mechanism hyperploidy, and this mechanism is called as hypoploidy. Hypoploidy, right? So hypoploidy matlab loss of the number of the genes, and hyper matlab addition of the genes it is. That is the numerical aspect. So here, apart from that, right? So there are the various factors which can bring the mutations in the plants. Various factors which can bring the mutations in the plant. So that's mean here we are having the domestication of the species. So whenever you are having the domestication breeding technique, right? In the domestication, we are converting the wild one into the cultivated form. In that case, sometimes there is a changes in the plant, either with respect to the genes or phenotype characteristics can be possible, right? So one is the domestication of the plant species, and Apart from that, whenever you are having the introduction of the exotic species, in the native area, sometimes there is a desirable changes. Some changes can be possible. Then, please write down the table. Then we are having the migration of the species. So whenever you are having the migration from the one place to the another place, over the period there is a changes can be possible. Then isolation of the organism. Best example, we are having the word we call as endemic species. What do you mean by endemic species? Endemic, they are confined to the only particular ecosystem. They are not available in the mainland. They are confined to the only particular ecosystem due to the some factors. It is. For example, we have seen that one tribal Centilage tribe in the Andaman and Nicobar, Centilage tribe. They are confined to the particular region. They are the endemic species in generally. Okay, due to the various factors. Then, whenever the organism is exposed to the radiation over the period, changes can be possible, and chemical agents due to which that development of the induced mutant varieties can be possible. Then natural forces. Then we are having the genetic drift. Then genetic erosion. And gene load. Right? So because of these factors, some are the biotic factors, some are the abiotic factors. So both the factors collectively can lead to the changes in the DNA sequence can be possible. For example, today morning we had earthquake. Earthquake can able to bring the some changes or not? It can. Did you fail? No. Right? So these are the various factors can lead to the changes in the DNA sequence can be possibilities. So here, look at the body. So we said changes. It can be structural or numerical. According to that, so it can lead to the new allele formation can be possible. Based on that, the new mutant varieties are developed in the agriculture. It is. So here, now let us look at the definition for the mutations here. So it is the change that occur in the DNA sequence due to the environmental factors. Not only environmental factors, it includes both biotic and abiotic factors here. So it is a process that generates the phenotype that differs from the normal one. Whatever the recombinant chromosomes we got, that recombinant chromosome is totally different from the normal chromosome. It is normal chromosome, and it results in the changes in the sequence of the DNA can be possible. That change either it can be addition or deletion or duplication or inversion or translocation of the chromosome can be possible. Then, in other words, it is a sudden heritable changes in the DNA sequence, right, or genetic material of the cell. Genetic material of the cell is called as mutation. Example, we can take addition mutation or duplication mutation. We can quote here, right? So here having these are the few examples we have A, B, C, D, and one is the deletion type of the mutations. Another is the A, B, C, D. Here we are having the B is reflected two times. Duplication. Another is the inversion type of the mutation. So one is the original chromosome. Another is the recombinant chromosome. Recombinant matlab whatever the result we are getting, that we called as recombinant chromosome. Resultant chromosome or recombinant chromosome, both the terms are same. You have to remember original chromosome and recombinant chromosome. Okay, the terminology is important. Okay. So here mutation, we said either it can be natural forces, either it can be induced factors. With the help of the human involvement also, induced mutation can be possible. So we are having using the radiation, antibiotics, and other kind of the alkylating agents. With the help of this also, induction of the mutation can be possible. Another is the natural mutation. Natural matlab spontaneous mutation, another is the induced mutation. We are having the two type of the mutation based on the occurrence. Based on the occurrence, one is the natural based, another is the chemical induced mutation. It is. So again, so for example, induced mutation can are used for the increasing the genetic diversity. We have used the radiation, we have discussed in the mutation breeding, right? Based on the chemical and radiation. We are having the radiation, another is the chemical. According to the such kind of the mutagens, right? We have increased the genetic diversity, then improved the crop production and preserving the natural resources. So some example of the plants that have been improved through the mutation breeding includes, for example, these are the some examples we have. So development of the improved varieties in the case of the rice in the Vietnam, Thailand, China and United States, and durum wheat, the durum wheat in the Italy and Bulgaria, the barley in the case of the Peru country and European nation, then soybean in the Vietnam and China, then leguminous food crops in the case of the Pakistan and India. So based on the radiation and chemical, we developed the some kind of the mutant varieties in the following the crops it is. So we said that following the factors. So we said that one is the chromosomal crossing over. Right, it is a mechanism. Then genetic drift in the ecosystem. Drift matlab earthquake is the one factor we can say, right? In the ecosystem can lead to the changes in the organism can be possible. Then exotic species and migration of the organism from the one ecosystem to the another ecosystem. Volcanic eruption can bring the mutation or not? Can be natural forces we say. Then gene load. Whenever you are having the overpopulation, sometimes changes can be possible. Then use of the radiation and UV, X-rays and gamma rays, we call it as radiation breeding. Then use of the mutagenic agents in the laboratory. We said ethyl methyl sulfonate chemical, EMS solution. Then and with the help of the hybridization work, mutation can be possible or not? Hybridization work like backcross method. With the help of the backcross method, also changes in the DNA sequence can be possible, right? Then we are having the isolation of the organism, then natural selection and domestication of the plant species. So with the help of these factors, there is a mutation can occur in the given ecosystem. So surely we can expect 15 to 20 marks question. What you understand by write down question. What do you mean by mutation? What do you mean by mutation? What do you mean by mutation? Question mark. How does it occurs in the nature? How does it occurs in the nature? Nature? Question mark. Describe the mutation. Describe the various type of the mutation. Describe the various type of the mutation. And its application. The various type of the mutation and its application. In the plant breeding. So 15 to 20 we can expect this question. Okay. Another possible question. Explain in brief mutant varieties. Explain in brief about the mutant varieties. Explain in brief about the mutant varieties. In brief about the mutant varieties. About the mutant varieties. Okay. See, so these are the two possible questions can be possibilities. Okay. So here, see, within the classification there are the different criteria we have to say the types of the mutation. So based on the lethality, these are the four type of the mutation can be possible. One is the lethal matlab all the alleles are die in the lethal condition. What all the 100% allele is going to die in the lethal condition. Another is the sublethal mutation. So in the sublethal mutation, we are having the 50% of the allele is going to survive in such situation. Third one, vital mutation. Vital matlab 100% of the allele is going to live, and subvital matlab more than 50% of the allele is going to survive in the such condition. It is a condition. In that cases, we are having the different composition of the allele survival can be possible. 100% nil and 50% and other more than 50% chances we have based on the lethality. Based on the lethality, right? That is the one classifications we have. Another one based on the direction. So going from the backward mutation. Backward matlab mutation from the present form of the character to the wild form. Going from the present to the past is called as backward mutation. So going from the present to the past can be possible or not in the nature? Can be possible or not? Can be possible. And so for example, we are having the cultivated species converting into the wild form. Another one, forward mutation. Forward matlab going from the past to the future. Past to the future. So mutation from the wild type to the new variety. So we are having the both backward mutation or forward mutation based on the direction. Right? There is the second classification. Third one, based on the degree of the effect. Effect matlab we are having the one is the micro mutation, another is the macro mutation. Micro matlab smaller changes. Smaller changes are observed in the phenotypic expression. And macro mutation matlab larger changes in the phenotypic expression in the recombinant chromosomes can be possible. That is the meaning it is. Okay, macro mutation, another is the micro mutation, another is the backward mutation, another is the forward mutation. Then based on the mode of the occurrence we said one is the spontaneous mutation, another is the induced mutation. Spontaneous matlab it occurs due to the natural forces. Due to the environmental forces and the plant mutation can be possible. Another is the induced mutation, right? Within the mutation due to the use of the physical agents. Physical agents matlab some are the radiation, some are the chemicals are used in the laboratory to make the mutant varieties it is. So again, so fifth one based on which issue. Okay, within the mutation we are having the some are the asexual mutation, some are the sexual mutation. Sexual it is also called as germinal mutation. Asexual it is also called as somatic. Within the sexual mutation again we are having the structural mutation, another is the numerical. Beside addition, deletion are the structural part. Deletion numerical matlab polyploidy and aneuploidy conditions here. That is the fifth classification here, right? Then sixth classification based on the place of the mutation, where the in which place mutation is going to take place, right? So one is the chromosomal mutation. So it occurs in the nucleus. Chromosomal mutation is going to take place in the nucleus, and it includes example we call as deletion type, duplication, inversion, and translocation type of the mutation can be possible. So all this mutation is going to take place in the nucleus region of the given plant cell. Another one, gene mutation. Changes in the base sequence of the DNA that alters the individual gene. For example, we are having the point mutation. Point mutation, it means the substitution of the one nucleotide in the given sequence. Substitution of the one nucleotide in the given sequence. For example, we are having the original sequence AAA, TCG. But in the recombinant, A is replaced with the C, adenine and cytosine. Point mutation, right? Substitution of the one nucleotide in the place of the another one. So cytosine is replaced with the adenine, right? That we call as point mutation. Point mutation in the given DNA sequence. Then third one, extra chromosomal mutation. It occurs in the mitochondria as well as in the chloroplast region in the plant cell. So sometimes it is also called as cytoplasmic mutation. It is. And seventh one based on the mutagens, right? One is the physical mutation genes, right? So we are using the alpha, beta, and X-ray to make the changes in the chromosomes, right? Another is the chemical mutation gene. Use of the chemicals such as alkalizing agents. We are having the ethyl methyl sulfonate solution, then acridine dry. Other chemicals are used to make the changes in the sequence. Do you remember in the plant breeding, in the mutation breeding, we have discussed the procedure, exposing the planting stack material. Then after that, we are having the mutation breeding 1, breeding 2, breeding 3. Do you remember flow chart, right? Then eighth classification, right, based on the functional mutation. See, so till the sixth, seventh classification, till the seventh are the most important one, right? In the eighth one, we are having the nonsense mutation. Nonsense mutation, matlab a genetic mutation that causes the premature stop codon, premature stop the codon to appear in the DNA sequence, resulting in the shortening and potentially non-functional protein production can be possible. So that we call it as nonsense mutation, right? Another one, the missense mutation. Missense matlab by mistakenly coding for the another amino acid, coding for the amino acid. So that means DNA changes that replace the amino acid in the protein with the different amino acid. That means code for the different protein, in the given genetic. See, in the life science we are having the genetic coding system, right? And there you will be having all the coding. It is a scientific process, but sometimes there is a missense of the coding can be possible. Okay, then we are having the silent mutation, changes in the codon but code for the same protein. Another is the frame shift mutation in the organism can be possible. See, these are the various steps we have in the case of the functional mutation, right? So these are the 8 classifications we have. So 10 marks we can expect classification of the mutation based on the various criteria. Some are the backward mutation, some are the forward mutation, some are the macro mutation, some are the micro mutation, some are the sexual mutation, some are the asexual mutation, and based on the lethality we are having the lethal, vital and subvital mutation can be possible. These are the 8 criteria we have to define the mutation. Okay. Then apart from that we are having the important features of the mutation. We said that it is a more of universal in the nature. In all the living organism mutation can be possible. So mutations are universal in the nature in all the living organism it is, right? Then it is subject due to the induced or environmental factors. So both the factors can bring the mutations in the given organism it is. Then it can lead to the variation, and which is basic ingredient for the evolution of the newer species, because it is highly required in the evolution of the newer species in the ecosystem. Then frequently they are deleterious in nature. And also they are in the recessive mode character. Remember, whatever the mutation changes we have in the given DNA sequence, they are not dominant character. They are always recessive. Most of the time they are always in the recessive character, but not dominant. You have to remember it is. Okay, whatever the changes we are having the recombinant chromosomes, right? In that cases, we are having majority of the recessive genes followed by the dominant gene. Okay. Then the mutation can occur at the any stage of the plant life cycle. Then mutation may be sexual or asexual in the nature. Mutation can be either lethal or non-lethal in the nature. It occurs both at the internal matlab genes of the plant, and external matlab peanut outside the plant characters. Okay, internal and externally occurring in the nature. Then it favors the organic evolution of the species and the ecosystem can be possible. Then it helps in gradual changes in the gene frequency of the population can be possible. So these are the various features we have in the case of the mutation. Then other features from the mutation. So we said in the mutant plant, changes in the leaf color can be possible. For example, in the ornamental plants, whatever the plants we have some yellow color, red color. Have you seen the ornamental plants? Duranta goldiana, Calipa, zebra plant, alag color hote. Okay, na? It's all result due to the induced mutations. You have to remember. Okay. Then appearance totally different. Phenotype is totally different with the normal chromosome. So whatever in the resultant plant we are having the different color of the flowers or biomass can be possible. Then natural occurrence. So it is naturally occurring in the nature. Sometimes whatever the naturally occurring mutations we have, sometimes it is also called as sports mutation or break mutation. You have to remember. Okay, break mutation, natural mutation is also called as, you can write down, naturally occurring mutation. So sometimes it is important. Naturally occurring mutation is also called as the sports mutation or break mutation. Is also called as sports or the break mutation. Also called as the sports or the Briggs mutation. The Briggs mutation. Right? That is the one feature sphere. Apart from that, right? Look at the body. We are having the whatever the changes we are getting, it can be reversion, can be possible. We said forward mutation and backward also can be possible. And so plants are usually revert back to their original form, can be possible in the following in the area, can be possible. That is the beauty we have in the given mutation. Then it supports the mutation breeding for making the variability in the plants. Then genetic adaptation. Mutation can help the plants to adapt to the changing environmental condition, because at presently society is changing, right? Environment is changing day by day. According to that, so in order to make the adaptability to the changes, so mutation is a basic ingredient factor, basic ingredient factors in the ecosystem. Then it brings the genetic variation, it brings the genetic variations in the plants. Then with the based on the such variation, evolution of the newer species can be possible. See, in the ecology we are having the word we call it as speciation. Any idea what you mean by speciation in the ecology? Development of the new species called as speciation. So in order to support the speciation, the mutation is very important. Okay, please write down. So speciation, it means the evolution of the newer species. The speciation, it means evolution of the newer species. In the ecology we are having the terminology we call it as speciation. It means evolution of the newer species. Evolution of the newer species in the ecosystem. Evolution of the newer species in the ecosystem. The ecosystem. Ecosystem. Right? So here, apart from that, there is the point transformation can occur in the point transformation. Matlab we said na in the given sequence when the single base pair is added or eliminated or changed in the genome can be possible. We said point mutation. Point mutation. C is replaced with the adenine. Cytosine is replaced with the adenine. Okay. Then we are using the various kind of the mutagens, right? So it is also the ecological factor that cause the mutations. It is. So here, apart from that, we are having the significance of the mutation, because in the plant breeding already we have discussed the 10 marks question. Mutant variety, it is a output of the mutation breeding. So along with that point, you have to add this point also. So mutation breeding is performed in the sugarcane, where sugarcane bud. Have you seen the sugarcane buds? Nodes hote na? So the nodes are exposed to the different kind of the radiation to generate the mutant varieties. Example, we have developed the HM658, HM661, Coimotor 213, Coimotor 602, and Coimotor 612. They are the name of the mutant varieties in the case of the sugarcane. One, mutation breeding is performed even in the case of the cotton crop. Example, the Malwal cotton variety are exposed to the X-ray to develop the indoor 2 variety. Indoor 2 variety are developed from the treatment with the Malwal cotton variety. Okay. Other varieties in the cotton include MA2, H190, 320F, another is a H14. Then mutant varieties are developed in the case of the wheat crop. Example, NP836, NP799. Mutant varieties are also developed in the case of the paddy, P500, P28, T1145. Then mutation can lead to the speciation. That means evolution of the newer species can be possible in the ecosystem. Then it causes the variability, improvement of the existing cultivar, right? So there is a genetic resource is increasing or decreasing? Increasing. Then mutant varieties are developed even in the case of the potato crop, as well as in the importing the new foliage color in the case of the ornamental plants. So in order to provide the new color in the biomass of the ornamental plant, mutation breeding is commonly practiced in the agriculture. That is the applications we have in the case of the mutation. Other application, right? It is a cost effective compared to the other breeding technique. With the lesser cost, development of the mutant varieties can be possible. Then very quicker method, within the shorter period, development of the variety can be possible. But in the case of the conventional breeding, we have to go for the 7 to 8 generation. है ना? But here within the एक बार ट्रीटमेंट करने के बाद में जल्दी हम कैरेक्टर चेंज कर सकते हैं, है ना? That is the one advantages we have. Then according to the experiment, they are saying it is a non-toxic, non-hazardous to the ecosystem they are saying. Okay, then environmentally friendly technique, then it reduces the breeding time from the 7 to 8 generation to the 1 generation or maximum 2 generation. Then they are called as non-transgenic. Is the transgenic or non-transgenic? Non-transgenic. Okay, but in the case of the transgenic crop, some kind of the standard guidelines will be there for releasing the any variety for the commercial purpose, right? So varieties developed through the mutation breeding are not transgenic. That is the reason they are not non-hazardous in the nature. So they don't have to comply with the same laws as transgenic plant. But in the case of the transgenic plant, standard scrutiny will be there. Now easy new, but in the case of the mutation breeding, it is an easy process for the implementation of the variety at the commercial level. Then it contributes to the global food security, because mutation breeding also is adding the various varieties in the case of the cotton, sugarcane, rice, potato, right? So directly or indirectly it enhances the food security for all the growing population, right? Then it improves the crop characters with respect to the yield, cultivation time, resistant to the disease, pest and climatic changes. So here, apart from this, so we are having the demerits of the mutation. So it can cause the abnormalities in the species, sometimes can be possible. Abnormalities can be possible, and it causes the genetic disorders such as albinism and hemophilia, which can be inherited to the progeny, can be possible. Sometimes induced mutant variety can destroy the original gene combination, right? Original gene combination matlab parental gene combination is having superior character, you know, with the involvement of the mutation, that superiority can be decreasable. So sometimes we are having the loss of the original genotype can be possible. Best example, if we can take brinjal, हमारा local वाला. Have you seen the wild variety of the brinjal? गाँव में इतना बड़ा था ना, हाँ अभी लंबा वाला आता है मार्केट में, है ना? लंबा वाला is hybrid, it's a cultivated one, but wild one day by day the character is decreasing, है ना? But in the पुराना time में हमका round वाला मिल रहा था brinjal में, but now we are having the लंबा वाला. There is a basic difference here, right? That is the best example for the loss of the original characters in the given properties, right? Then induced mutant varieties can affect the natural process in the evolution of the newer species, can be possible, right? So because with the help of the chemical process or radiation process, we are involving the ecosystem, we are interfering in the ecosystem process. Okay, so not today, but in the long term there is a limitation can be possible to all the stakeholders. It is okay. So other limitations from the mutation, it can be harmful to the organisms in the long term. Then low frequency of the disease. Low frequency matlab, for example, we said original chromosome and recombinant chromosome. In the recombinant chromosomes, we are getting only low frequency of the desirable character. Only frequency is very less. And within the less also, we are having the most of the genes are recessive gene than the dominant character. Okay, that is the reason we are getting the low frequency of the desirable characteristics. Then difficult to detect the recessive mutation. Whatever the recessive we are getting, sometimes it's difficult to detect the recessive gene compared to the dominant. Then large dose of the mutation at the chemical and radiation level, we require the large dosage of the chemical to induce the character in the plant variety is required. Sometimes whenever if you go for the larger doses, sometimes it can be having the demerits can be possible, right? Then undesirable side effects over the period of the time can be possible, and over the period there is a chimerism. That means irradiation of the multicellular structure. Whenever you are having the use of the radiation in the given structure or mutagens, can result in the chimerism in the plant character can be possible. Chimerism, best example, have you seen the maize? एक क्रीमी कलर मेज होते हैं, समटाइम रेड कलर मेज होते हैं, एक बार उसमें देखा था एक पार्ट में सभी नॉर्मल वाला क्रीमी येलो कलर में होते हैं, उसी बीच में एक रेड कलर और पिंक कलर में होते हैं. There is a chimerism can be possible. See, so these are the few things we have in the demerits of the mutation. Apart from that, so one is another is the reduced survival. Whatever the mutant varieties we are developing over the period, survival will be very less. Correct my point. Survival, it's not, they're not having the stable survival for the longer period of the time. Whatever the varieties we are getting in the mutant category. Then, no significant changes in the traits. Induced mutation may not always result in the significant changes in the certain traits. May not always. Okay. Then, biosafety effect sometime we are speaking, but it is we said in the previous slide it is a non-hazardous, but sometimes there is a biosafety issues can be possible because we are using the mutation with the larger dosage. Over the period, some kind of the limitation can be possible. Then subpar performance. Whatever the mutant varieties we developed, the performance is not up to the mark. Is not up to the mark. So always we are getting the below the average performance in the developed mutant varieties. That we call as the subpar performance in the mutant varieties here. So these are the some limitations we have, right? See, so these are the few things we have today. So maximum 15 to 20 marks we can expect in the case of the mutation breeding case. Okay, it's all today, and tomorrow we'll be having class.